Swayam Prabha. Digital India. Educated India.
Hello everyone. Welcome to the fifth and final lecture of fourth unit of plant biotechnology course. In this unit, we have learned till now about the plant and microbial interactions, plant growth promoting rhizobacteria, nitrogen fixation cycle, and the mechanism of nitrogen fixation. And now we are learning about the various methods of plant genetic engineering. So today we will continue talking about the plant genetic engineering and its applications. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University, New Delhi. In JNU, I teach plant biotechnology and genetic engineering courses at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. This project has been sponsored by DTH Swayamprabha, MHRD, New Delhi. So in the last lecture, we talked about the agrobacterium disease biology, and today we will continue talking about agrobacterium as a tool for plant genetic engineering. I will talk about the construction of cointegrate and binary vector system that are used with the agrobacterium, followed by a step-by-step tomato transformation. I will also talk about the methods used for molecular characterization of the transgenic plants, and at the end, briefly I will discuss about the applications of plant genetic engineering. So we learned about the mechanism of the agrobacterium action, and now the question is that how can we use agrobacterium as an engineering tool for transforming plants. If you carefully study the mechanism of agrobacterium infections, you will find that right from the sensing of the plant phenolics by agrobacterium and its colonization on the cell surface, to the integration of the tDNA into the plant genome, which includes several steps like induction of the virulence genes, processing of the transfer DNA molecule, formation of the communication channel, transfer of the tDNA molecule from the agrobacterium to the plant cell, and integration of the tDNA in the plant genome. So tDNA is acting a passive entity through all these steps. That is, it is not participating actively in these steps. In other words, tDNA do not code for any component that are required for all these processes, that is from the sensing till the integration. All the components that are required for this purpose, they are encoded by the virulence region of the TI plasmid. And there are some other components that are coded by the bacterial genomic DNA that may not be essential for the infection, but they improve the efficiency of the infection. So tDNA encoded genes start to work only after the tDNA is integrated into the plant genome. And now they synthesize the opines and the plant hormones, as the tDNA has the genes for these components. Opines are used by bacteria as a carbon and nitrogen source, and the plant hormones causes the actual disease, that is the Crohn galls, by stimulating the uncontrolled cell division. Therefore, the tDNA encoded components do not have any regulatory role in the preparation of the tDNA and transfer of the tDNA from the agrobacterium cell to the plant cell, and even its integration into the plant genome. And this property of the agrobacterium tDNA biology can be used as a tool to transform the plants. In the last lecture, we talked about the TI plasmid and its role during the infection process. So TI plasmid is essential for the transformation. These are large plasmids, that is their range is somewhere around 200 kb or even larger. They have two major components that participate in the infection process, that is the virulence genes and the tDNA region. tDNA is actually transferred to the plant genome, and virulence region, it codes for the factors that are responsible for this transfer. Now just think, if the tDNA is not playing any role in its preparation and the transfer to the plant cell for integration, how does the bacteria identify that this is the tDNA segment that has to be transferred to the plant genome? Why doesn't it select any other component of the TI plasmid or the bacterial genome? And the answer is the presence of these two border sequences, that is left border and the right border. So the virulence protein or enzymes which are coded by the virulence region or the Vir genes in the virulence region, they recognize these left and right border sequences and prepare the copy of the DNA segment which is present between these borders. So therefore, it does not matter what kind of DNA is present between these left and right borders, and there is no mechanism or the validation step that tDNA should contain the gene encoding for the opines or the plant hormone synthesis. So even if we replace the tDNA segment with some other DNA, it will not affect the transfer process as long as left and right borders are intact. So basically, we engineer the tDNA on the TI plasmid and put it back in the agrobacterium and let the agrobacterium transfer this tDNA to the plant genome for us. So the first step is the vector construction. Now we cannot use the TI plasmid as such to transfer the gene of interest to the plants. There are several important steps, like first we need to disarm this vector, that is we need to remove these genes that encode for the plant hormones or the opine synthesis. Further, we need to develop methods to clone our genes of interest in this particular tDNA region. Many other DNA segments that are unnecessary on the TI plasmid, they can be removed from these plasmids, like the opine catabolism genes. It will help to reduce the size of the plasmid, and smaller plasmids are usually easy to manipulate and handle. So depending upon the strategy of engineering TI plasmids, vector constructs are grouped into two categories. One is the cointegrate vectors, and second is the binary vector system. So the first category is the cointegrate vectors. Now cointegrate vectors are the deletion derivatives of the TI plasmids. So as we discussed earlier, that we simply replace the tDNA segment of the TI plasmid with desired DNA segments that we want to transfer to the plant genome. And further, some of the other components that are unnecessary for the tDNA transfer function, they can be simply removed from the TI plasmid, like these opine catabolism genes which do not play any role during the tDNA transfer process. They can simply be removed from these plasmids. Therefore, the final vector would have only those components that are required for the multiplication of the plasmid, that is for its survival, and the components which are required for the agrobacterium infection, that is the virulence and the tDNA transfer. In order to construct these vectors, first the TI plasmid are disarmed, that is the removal of the opine and plant hormone synthesis genes from the tDNA region. However, other required sequences are introduced in this region which can be later used for cloning the desired DNA segments through the homologous recombinations. So the final vector would look something like this. It has the virulence region, other components that are essential for its multiplication. Then it would have left and right border and the newly inserted sequences between the left and right borders. This particular vector is an example of PGB series vector where opine and hormone synthesis genes have been replaced with a small segment from PVR322 vector. Now PVR322 vector is a cloning vector which is small in size and has a high copy multiplication in the bacterial cells. The desired DNA that is to be introduced into the plant is cloned in the conventional E. coli plasmids. Here in this case it's the PVR322, and these are also called as the intermediate vectors. These are small in size and they are easy to manipulate. However, these vectors are incapable of the replication in agrobacterium, and also they lack the conjugation function. So they are just the cloning vectors. So once the gene of interest is cloned in this plasmid and other required sequences like a selection marker or the reporter marker, if we are including it on the tDNA, these smaller vectors are integrated into the disarmed GI plasmids. And for this integration, these homologous sequences are required as this integration occurs through the homologous recombinations. So these sequences which are present between the left border and right border, they recombine with the corresponding homologous sequences in the cloning vector and are used to insert this smaller vector as an insert in the disarmed GI plasmid. This creates the cointegrate vector for the plant transformation. These larger disarmed TI plasmids are in the agrobacterium, and therefore for recombination to happen, these smaller plasmids with the desired DNA need to be transferred into the agrobacterium. For this purpose, another helper plasmid that has the genes required for the transfer and mobilization of the plasmids can be used. These helper plasmids help in the transfer of these conjugation deficient intermediate plasmids to the agrobacterium. And after the successful recombination process in the cells, these smaller plasmid segments, they are introduced into the disarmed TI plasmid, and it creates the cointegrate plasmids that has the desired DNA segment between the borders, and all the virulence operons are there. So this plasmid is capable to replicate in the agrobacterium, and when infect plants, it would transfer the tDNA, now it is the desired DNA, to the plants and transform the plants. So in this way, cointegrate vectors are constructed having both the virulence genes and the tDNA on the same plasmid. So cointegrate vectors, both the tDNA, that is the desired DNA to be transferred to the plant genome, and the virulence region, they are present on the same vector. These vectors have a selection marker for bacteria as well as the plants. That is, for the manipulation purposes, we need to multiply them in the bacteria or maintaining them in the agrobacterium. And for the plant selection, markers are introduced to select the transformed cells, and these are integrated into the tDNA, and they are used to regenerate the transgenic plants. TI plasmids are large in size, that is about 200 kb or even the larger, and therefore these plasmids are difficult to manipulate for the cloning purposes. In order to have successful recombination for the integration of the intermediate vector into the disarmed TI plasmid, long homologies are required between these two plasmids. And this further complicate the process and make the engineering more difficult. Further, these plasmids are low copy in number, and therefore their multiplication and large scale preparations are difficult. In addition, the required unique restriction sites may not be there, and therefore cloning of the fragments of the DNA segment directly in these vectors is difficult. So the second system is the binary vector system. So here is this is a cointegrate vector that has virulence genes as well as the tDNA that needs to be transferred. We also talked about drawbacks or the challenges associated with these vectors. So now the question is, how could we reduce the size of the plasmids and make the handling or the manipulation much easier? A key finding that the virulence genes which are involved in the transfer of the tDNA need not to be present on the same plasmid where the tDNA is there. Therefore, the virulence genes and the tDNA segments can be placed on the separate plasmids. This means we can have two vectors like this one, where one contains the virulence genes and the other smaller vector would contain the tDNA to be transferred to the plants. And the virulence protein encoded by the genes present in this vector can recognize the left and right borders present on the another vector and successfully can transfer the tDNA to the plant cell, as long as both of these vectors are present in the same agrobacterium cell. And this system of vectors where the virulence genes and the tDNA, they are present on the two separate vectors and they are used for transformation, is called as a binary vector system. So the binary vector system consists of two plasmids, that is Vir helper plasmid and the tDNA containing binary vectors. So this plasmid is a tDNA binary vector. It has a tDNA that is flanked by the left and right borders, and this other one is the Vir helper plasmid that carry the virulence operon having all the Vir genes that are required for the tDNA transfer. In the cointegrate plasmids, both the tDNA and virulence genes, they are present on the same plasmid, and the virulence genes, they act in the cis, that is the tDNA is present on the same plasmid. However, in this case, the virulence genes are placed on the Vir helper plasmid and they act in trans to recognize the left and right border which are present on the other plasmid. However, they should be in the same cell. So typically the gene of interest is cloned in the tDNA binary vector having left and right borders like this one. These vectors are smaller in size and hence they are easy to handle during the cloning and transformation process. Multiple unique restriction sites for the cloning are available, and even if these are not available, these can be introduced easily and another vector with the unique restriction sites can be synthesized. These vectors are high copy number plasmids, that is within the same cell. These plasmids can multiply and accumulate large number of their copies, and therefore they are easy to isolate and are not limited by amount of the plasmid for manipulation. Further, these are easily replicated in the E. coli. The Vir helper plasmids are prepared once and these are then placed in the agrobacterium strains and maintained there in these strains. So these vectors do not need any further manipulation for the cloning of the desired DNA segments. All the manipulations for the cloning of the desired DNA are associated with the tDNA binary vector. So we make the required manipulations, that is the cloning of the required tDNA segment in this small vector, and for successful expression of the desired genes in the plants, we need to provide all the required elements that are required for the gene expression in the plant cells, like there should be a suitable promoter, then the actual gene sequence should be there. There should be a suitable terminator that is required to terminate the transcription, and then the additional sequences like the enhancers or the signal sequences if we want to target the protein accumulation to the particular compartment of the cell. So once this vector is ready, we can easily transfer these vectors to the agrobacterium cells that already have the Vir helper plasmids, and now the agrobacterium having the Vir helper plasmids and the tDNA binary vector is ready for the transformation process. So in this way, manipulation in the binary vector system for the cloning of the genes is much less as well as easier too as compared to the construction of co-integrate vectors. This is an example of actual tDNA binary vector, that is the pCambia GOI. Now this pCambia is a series of plant expression vector where different kind of a promoter or a terminator and other regulatory sequences combinations are there. So in this case, here is the left border, and from here till the right border, this whole segment is the tDNA. Here in this tDNA, we have 3 expression cassettes. One is the gene encoding for the antibiotic, that is the kanamycin resistance gene in this case. Second is the gene of interest that we want to insert or that code for the actual trait. And the third one is the gene encoding for the reporter protein, like the Gus enzyme in this case. Now, in order to express these genes in the plant system, we need to provide the other required sequences like the suitable promoter. Promoter derive the expression of the gene, and further their activity may differ in the different plant species or even among the different tissues within the same species. For example, many promoters that have a high activity in the dicots may not have the same level of activity in the monocot plants. So depending upon the species or the tissue where we want to express the candidate genes, accordingly suitable promoter is selected. Similarly, other required sequences such as the transcription terminator, which is required to terminate the transcription process, like here NOS terminator in this case and 35S terminator in this case have been used. In addition, enhancer sequences which are used to increase the activity of the transcription, or the signal sequences which are required for targeting the expression or the accumulation of the protein in specific compartment of the cell, they are provided on this expression cassette. Now let's learn about the agrobacterium mediated plant transformation step by step. First step is the preparation of the suitable vector and the transformation of this vector in a suitable agrobacterium strain. The infectivity of the agrobacterium varies among the different plants, and therefore several different types of agrobacterium strains having different level of infectivity have been prepared. Therefore, we need to select a suitable strain of agrobacterium depending upon the candidate plant that we are going to transform. Next is the required culture media components. So typically we need to have an optimized in vitro regeneration protocol available for the plant species that we are transforming. So all the media components that are required in that regeneration protocol, they should be available. However, if the regeneration protocol is not available for that species, first we should optimize an efficient regeneration protocol before going ahead with the transformation. Next step is the explant preparation. As we discussed earlier also about the explant selection in the first unit. there are several different types of the plant cell tissues or organs that can be used as an explant for regeneration. Theoretically, any of the plant tissue or the cell which is living can be used as an explant. But mitotically active tissues like the immature embryos, shoot tips, meristems, or young leaves that are more successful for callus induction. So the suitable explants for which the regeneration protocols are available are selected and prepared for the transformation process. Now the explants are infected with the agrobacterium strain having the desired plasmids, and the process is called as a co-cultivation. This is followed by the selection of transformed cells, and usually it is performed using the antibiotics for which the required antibiotic marker genes are introduced along with the tDNA. This helps to select the transformed cells, and they are allowed to grow and form callus. Later, using the required hormonal combinations, morphogenetic responses are initiated and shoots are regenerated. Once the shoots of reasonable size they grow, they are transferred onto the rooting media where these will be induced to form the roots. And once these shoots form the roots, these plantlets are transferred to the pots for hardening. Let's have a quick look on the various steps of the agrobacterium mediated transformation in tomato. Here in this example, cotyledons have been used as an explant from 8 to 10 day old tomato seedlings. So we sterilize the tomato seeds and germinate them in the aseptic environment. As these seeds are grown in the aseptic conditions, these will be free from the microbes, and further sterilization process during the explant preparation would not be required. So these first leaves that appears when the tomato seeds germinate, these are called as the cotyledons, or these are also called as embryonic leaves. These cotyledons are excised from the tip as well as on the base of the leaf and are placed on the MS media supplemented with the required hormone for the induction. These explants are usually cultured on this media for about 2 to 3 days before they are actually infected with the agrobacterium, and this is called as a pre-culture. During this time of pre-culture, the explants they recover from the stress caused by the cutting injury and separating them from the parent plant, and they also adapt to the culture condition and also induce the cell division. After the explants are pre-cultured on the pre-culture medium for about 2 days, it's time for the agrobacterium infection, which is called, that is the co-cultivation. For this purpose, all the pre-cultured explants are transferred to the agrobacterium solution in a Petri plate or other suitable container, and these are incubated together for about 5 to 7 minutes in this solution. After about 5 minutes, explants are removed from the agrobacterium solution. Extra liquid medium is blotted from these explants and they are transferred onto the co-cultivation medium. These are incubated on this medium for about 2 to 3 days depending upon the type of the agrobacterium strain, that is the infectivity level of the agrobacterium strain, or the type of explant used. And this incubation is called as a co-cultivation. When the explants are blotted to remove the agrobacterium solution, it does not remove all the bacteria. There will be sufficient number of bacteria that stick to the explants which will grow with the explant and establish the infection, and during this process they will transfer the tDNA to the cotyledon cells. However, if the extra solution is left with the explants and they are placed on the co-cultivation media, it can compromise the explant health and the explant may degrade. Next step is the selection of the transformed cells and the callus formation. So after 2 days of the co-cultivation, the explants are washed thoroughly with the sterile water and they are transferred onto the selection media. These are incubated in the controlled condition of light, temperature and humidity, and they are allowed to grow there. Now the selection media specifically contain the antibiotics for which the antibiotic marker genes have been introduced through the tDNA into the plant cell. And this will help to select the transformed cells, as only those cells would be able to grow on the selection media that would have been transformed, that would have a tDNA integrated into their cells, and they will be expressing the antibiotic resistant protein in these cells. So if we just zoom in this particular selection plate, we can see that the development of the calli on the cut end of the cotyledons. These are healthy calli and these are growing on the antibiotic selection media. Whereas if we see this other part of the cotyledons, that is the part where we can see the tissue is turning dark or creamish, it is losing its green color slowly and it is not growing also. These are the untransformed cells or the tissues that are dying on the antibiotic selection media. Now, once the sufficient amount of the callus is formed, these cells, these calli are transferred onto the regeneration media to induce the morphogenetic responses. The regeneration media usually contain required combination of the plant hormones for the induction of shoot regeneration, like in most cases it is the balance of the auxins and the cytokinins that is crucial for the regeneration of shoots. So these shoots, when they grow to a suitable size, they are excised from the parent calli and they are transferred onto the rooting media. In a few days, these shoots will regenerate the roots, and now the plantlets are ready for their transfer to the soil in the pots. Now, in many cases, plain MS media is enough for the root induction. However, in many species, low concentrations of auxins are required for the induction of roots. So these plantlets are removed from the medium. Roots are washed and plantlets are transferred to the soil. In order to maintain the humidity or save the plants from sudden shock of the dry environment, either these pots are placed in the humidity regulator chamber, and if that is not available, we can simply cover these plants with the polythene bags. Once the plant acclimatizes to the soil within a few days, these bags are removed and the plants are allowed to grow to maturity in the contained facility. So this is the whole process of tomato transformation, how we start with the cotyledon and raise the transgenic plants using the agrobacterium mediated transformation. Next step is the molecular characterization of the plants. Once the transgenic plants are transferred to the pots, these are ready for the molecular characterization or the validation of the integration or the expression of the transgenes. There are various steps of the plant transformation we just studied, that is the selection of the foreign gene to be transferred to the plants. Then is the cloning of this gene and construction of a suitable vector for the transformation. Transfer of the vector to the agrobacterium, followed by agrobacterium mediated transformation of the plant tissues, selection of the transformed cells and the callus induction, and next is the shoot regeneration from the callus, followed by the regeneration of the transformed plants by the induction of roots on the shoots. Finally, the plants are transferred to the soil for an acclimation process. Now, very little tissue is required for the molecular characterization, and it can be actually performed even at the earlier stages of the transgenic plant development. We need not to actually wait for the plant to be acclimatized in the soil to start the molecular characterization. Like even when the shoots are transferred onto the rooting media, little tissue can be harvested that can be used for the molecular characterization of these plants. Various steps of the molecular characterization of the transgenic plants are: first one is checking the integration of the tDNA into the plant genome. Then next is, if the gene is integrated, we need to check whether the tDNA is transcribed or expressed. Further, if the transgene is translated and the recombinant product is formed, or if the recombinant product formed is functional or active. So the first one is the integration of tDNA. Now, PCR amplification is most commonly used method for this purpose. And in order to perform the PCR, we would need two things, that is the template DNA and the gene-specific primers. Therefore, small amount of tissue is harvested from the putative transgenic plants, and the genomic DNA is extracted from this tissue using the routine procedures. The genomic DNA isolated from these plants is used as a template for the PCR amplification. And the second part is the primer. So here is the tDNA of the binary vector, and so if this vector would be used for the plant transformation, the DNA segment from this left border to the right border is expected to be transferred to the plant genome. So theoretically, we can prepare the primers from any part of this tDNA segment for the PCR amplification. However, in order to make sure that the complete tDNA is inserted in the plant genome, more than one primer pairs can be used from the different parts of the tDNA. So in addition to the gene of interest specific primers, we can use the antibiotic marker or the reporter gene specific primers too for the PCR amplification. Now here is the picture of an agarose gel when the PCR products are electrophorezed. First lane is the marker that help us to identify the size of the amplified fragment. However, in this case, it's not helping to identify the size, as we do not have any marker band in this particular region. And therefore, another marker have been used in this case in the last lane that would help us to identify the smaller PCR amplicons. So therefore, we need to choose the appropriate markers to check if the expected size amplicons are amplified. Now, how would we know what is the expected size of the amplicon? Well, it will depend upon the primers. So when we design the primer from this particular tDNA region, we would know that where they are mapping and what is the distance between the two locations where these two primers are mapping, and this distance will be the expected size of the amplicons. The PCR amplification samples that are marked with the numbers 1, 2, 3, 4 here, they are from the transgenic plants. In addition to this, two controls are used, that is the positive control and a negative control. Now, the tDNA binary plasmid that was used for the plant transformation, it is used as a template for a positive control. Now, it means that it should amplify the expected size amplicon, because we know that the tDNA is a part of this plasmid that was used for the transformation, and we have designed the primer from this tDNA, and if we will use this plasmid as a template for amplification, it should certainly amplify the fragment, and that's why it is called as a positive control. Next is the WT, which represents the wild type plants. That is, the genomic DNA from the untransformed plants is used as a template for PCR, and because the tDNA would not be there in the untransformed plants, therefore PCR amplicon is not expected in this case. So we can see here that we got the PCR amplicon in case of the positive control, but did not get any amplicon in case of the genomic DNA from the wild type plants. And then in all the transgenic plants, we got the expected size of the amplicons. This is an example using one pair of primers. However, multiple primer pairs can be used for this purpose. So this confirms that the tDNA from the plasmid has been integrated in all these putative transgenic plants. After the confirmation of the integration, next step is to check if the gene is expressing or not. Integration of the tDNA in the plant genome does not make sure that the gene will certainly express. The location of the integration in the genome may affect the expression of the genes present on the tDNA. Like sometimes tDNA may be integrated into the heterochromatin region where the expression is almost negligible, or it may be integrated close to a repressor that will not let it express. And therefore, we need to validate the expression. RT-PCR and qPCR, that is the quantitative real-time PCR, these can be used for this purpose. Earlier, Northern blotting was used for checking the expression of the genes. However, as the PCR-based methods, they are easier, faster, and they produce better results. Therefore, these are more popular. For this purpose, we harvest a small amount of tissue from the putative transgenic plants and extract the total RNA using routine RNA extraction procedures. Here is the gel picture of the agarose with the total RNA samples electrophorezed on it. Again, the first lane represents the size marker, and the lane 1 to 5, these represents the total RNA isolated from the different plants. Now we synthesize the complementary DNA from the extracted RNA and use that complementary DNA or the cDNA as a template for the PCR-based expression analysis. However, we need to make sure that the RNA should not be contaminated with even little amount of genomic DNA. Now, what will happen if the genomic DNA contamination is there in this RNA? Now, if the genomic DNA contamination is there in the cDNA, primers may bind to the tDNA integrated into the plant genome and can amplify or give the amplicon. However, this amplicon would not represent the expression of the gene, rather it will show the integration of the gene in the genomic DNA, and therefore this would produce spurious results. Now, if we perform a simple RT-PCR, we have to analyze the PCR amplicons on the agarose gel. Like here are the results from the candidate gene-specific primer. Again, here the first lane is for the suitable size marker, and then the plasmid is for the positive control, and WT is for untransformed plants, and number 1 to 4 represents 4 different transgenic plants. This amplification will confirm the expression of the transgene from integrated tDNA. However, if we use the qPCR for expression analysis, we get the results in the form of numbers and amplification curve. So in this case, the fluorescent dyes are used, and machine read the fluorescence signals and quantify the amount of DNA amplified at each cycle of the PCR. So here we have two samples, that is sample 1 and sample 2. We have used each sample in triplicates, that means 3 reactions for the same samples were put, and here they were designated as A, B, and C. These numbers are called CQ or the CT values, and this is actually the representation of the expression. Higher is the number, lower is the expression of the target gene, and if lower is the CT value, it represents the higher expression of the target gene. So in this case, the expression of the target gene is much higher in the sample 1, where the CQ values are in 20s, and in sample 2 it is much lower as the values are in 30s. So in this way we can use the PCR, simple RT-PCR or the qPCR, to validate whether the genes or the transgene presence on the tDNA they are expressing or not. After the confirmation of the tDNA integrated and the genes, the transgenes present in the tDNA are transcribed, next step is to validate if the translation is occurring or not. That is, whether the protein is synthesized and accumulated in the cell, or if the synthesized product has been required activity or functionality or not. So many times though the transgenes are transcribed, however, they may not be translated. And this may be because of the presence of rare codons in the transgenes, or in several cases the recombinant protein is not tolerated by the host system and it is immediately degraded or cleared from the system. For this purpose, again the total protein is isolated from the tissues harvested from the transgenic plants, and the Western blotting or the ELISA based assay can be performed to validate the protein synthesis. However, we need to have the antibodies specific to the target proteins to perform these experiments. Further, if the transgene is translated and recombinant protein or the product is accumulated, it may not have required activity. That means it may not perform the required functions. Like it may not be folded properly and hence lose its activity. Or it may need some post-translational modifications for its functionality, and these may not be added by the system. Therefore, after validation of the accumulation of the recombinant product, these are validated if these have the required functionality or not. The validation procedure or the types of assays that are used to check the activity or functionality of the recombinant protein would depend upon what type of this protein is. So these are the standard steps for the molecular characterization of the transgenic plants. Next is the applications. Now, plant genetic engineering is about 40-year-old field and has developed rapidly due to the greater understanding about the biological mechanisms. The improvement of the existing plants for various agronomic characters, or introducing the novel characters in the ultimate goal for any plant genetic engineer. Let's briefly discuss about the various applications where the genetic modification either has made a significant difference or has a huge potential to make a significant impact. So the first one is the engineering stress tolerance. Now, one of the first such example is the engineering virus resistance in the papaya. At a time in 1980s and 90s, Hawaiian papaya, so Hawaii is an island state in the United States. The papaya here was on the extinction because of the papaya ring spot virus disease. All the traditional strategies failed to control the virus infection, and then the transgenic approaches was tested for this purpose. The gene encoding for the coat protein of the papaya ring spot virus was used to engineer resistance against the virus infection. This coat protein mediated systemic cross protection was very successful in this papaya infections. And similarly, the virus resistance has also been engineered in other plants like squash and potato. Considerable progress have been made in engineering tolerance to several other stress types such as the abiotic stresses, like drought tolerance in maize. Like the drought tolerance cultivars of maize has been engineered, and drought guard cultivar have been released. Similarly, Verdeca HP4 has also been released for the soybean, which is also drought tolerant. Next is the herbicide resistance. Now, herbicide tolerant crop varieties have been produced for several crops like maize, canola, cotton, lentils, soybean, sugar beet, and so on. So basically, herbicide tolerance varieties contain a transgene that provide resistance to one or more broad spectrum herbicides. The use of these herbicide tolerant varieties can provide a better beet control and improve the overall yield too. Several commercial cultivars of the herbicide resistant crops are available in the market, like Roundup Ready for the Medicago, and then in Vigor, Liberty Link, or the TrueFlex for the canola. Similarly, many other cultivars have been approved or in the process of approval, like the Navigative for canola, Glytol for cotton, and so on. And for more information regarding this, you may visit the GM, or a genetically modified organism approval database, which is hosted by ISAA, that is International Service for the Acquisition of Agri-Biotech Applications. Next is the insect resistance. Here, BT varieties are very popular. Basically, in these plants, new genes have been introduced that codes for insecticidal protein. These proteins is present in the soil microbes, and they have been used safely for over 4 to 5 decades organically to protect the plants from the harmful insects. Several insect-resistant plants have been produced, like we know about the BT cotton or the BT maize, which are grown on the large scale. Similarly, other crops like BT brinjal or the eggplant, flax, potato, tomato, coffee, etc. have been also engineered with the insect resistance and are in the process of approval. Another category is to improve the nutrition quality of the food product. And in this case, golden rice is one of the most popular event that we know about. Here is the pathway or the various enzymes that are required for the synthesis of the beta carotene, which is the precursor of vitamin A. Therefore, in order to synthesize beta carotene from zerenyl diphosphate, these enzymes, that is the phytoin synthase, desaturase, and lycopene beta cyclase, are required. However, in rice, all these enzymes are absent, and hence normal rice would not be able to synthesize beta carotene. Therefore, in rice, zerenyl diphosphate is the last product from the beta carotene pathway. And now, if we want the rice to synthesize the beta carotene, we need to provide these enzymes in the rice genome. So the gene encoding for these enzymes from the daffodil and bacteria, they were transferred to the rice genome through genetic engineering. The expression of these three genes complete the beta carotene synthesis pathway in the rice plant. Here in the second step, a single enzyme, it performs the function of both these enzymes, that is the phytoin desaturase and carotene desaturase, and therefore introduction of the enzyme completes the beta carotene pathway in rice. Now, these transgenic plants would be able to synthesize the beta carotene, and because of the synthesis of beta carotene, the color of the rice changes from the white to golden, and that's why it is called as a golden rice. Similarly, other traits like the reduction in the post-harvest losses or improving the growth and yield of the crops have been introduced through the genetic engineering in several plants. In fact, the flavor saver tomatoes were the first transgenic crops that were licensed for the human consumption. These tomatoes were engineered, or these engineered tomatoes had improved shelf life by staying firm and ripe for longer period of time. Similarly, genetically engineered cultivars have been developed for maize, soybean, or the eucalyptus, where the transgene expression result in the increased growth or the better photosynthetic efficiency, resulting in overall yield gains. Now, another aspect is the molecular farming. Molecular farming is another application of the plant genetic engineering where the significant progress has been made. That means plants can be used as bioreactors for the production of proteins or metabolites of the pharmaceutical or industrial importance. Now, it is important because the plant provides several unique advantages over the other bioreactor systems like the microbial bioreactors or the mammalian cells-based bioreactors. Large number of such products have been produced in the plants and several of them even have been commercialized. Broadly, these products can be categorized in 4 or 5 major categories. And the first one is like the therapeutic products. Several therapeutic products such as the antibodies, growth factors, replacement proteins, enzymes for the therapy, immune system stimulants or the suppressants have been produced successfully in the transgenic plants. Then is the nutritional components. These may be the proteins, amino acids, vitamins, minerals, fatty acids, carbohydrates, etc. And so either the existing concentration of the nutritional component can be increased or the new pathway can be introduced into the crops. Then next category is the vaccine antigens. Now the production of the vaccine antigen in plants gave the concept of the edible vaccine in 1990s. However, now it is more popular by the name of the plant vaccines. So basically, antigenic proteins or the peptides can be produced in the plant tissues and they can be used for the vaccination purposes. A large number of such antigenic protein for various pathogenic organisms have been produced successfully in plants. Then next is the biodegradable plastics where various types of polyesters have been produced in plants like the PHAs, polyhydroxyalkanoates or polyhydroxybutyrates. Then the starch-based polymers, they are the important compounds and they can be produced in the plants. Similarly, several proteins of the industrial importance such as the diagnostic proteins, research agents, enzymes for the brewing industry, textile, paper or the detergent industry are being produced in the plants and have been commercialized too. In this way, plant genetic engineering has innumerable applications in the various fields and has a huge potential to serve the humanity. So in summary, today we learned about the agrobacterium tumefaciens as a tool for plant genetic engineering. We talked about the construction of the Cointegrate and binary vector system that is used with the agrobacterium mediated plant transformation. I also discussed about the step-by-step transformation protocol for tomato and the molecular characterization of the transgenic plants. Finally, I briefly discussed about the various types of applications of plant genetic engineering. So this was the last lecture of this last unit of this course, and with this it completes the plant biotechnology course. Thank you.